Hi, everyone. We're going to finish this week, Wednesday and Thursday, with two shorter lectures that are going to cover DNA and gene expression. So, we're going to review DNA and go a little bit more in depth, looking at the DNA molecule and genes. We are then going to examine the process whereby genes are transcribed by mRNA molecules and then translated into functional proteins in the cell. And then we'll talk a bit about gene expression as well, because not all genes are expressed simultaneously. I think I've mentioned before that you have a copy of every single gene inside of every single cell of your body, and yet you're not making eyeballs on the palms of your hands, even though the cells in your hands definitely have the instructions for making eyeballs. So what regulates what turns genes on and genes off, and when and where does that happen? We'll talk about that as well. So the goals for today and tomorrow are to describe what DNA is and what it does. We will explain the process of gene expression and the collaboration of nature and nurture. We'll do that today. And then tomorrow we'll dive into the causes and effects of damage to the genetic code. So specifically, we'll be looking at mutation. Now, we've already talked about DNA when we talked about macromolecules, specifically that DNA contains instructions for the development and functions of all living organisms. And that, again, tells us something profound that we'll expand on when we get to the section on evolution. There's a reason why all living organisms share this molecule, this unique molecule that contains instructions for making organisms. It's because at some point in history, granted it was a long time ago, we all shared an ancestor that had DNA molecules with instructions for making organisms. So this tells us something profound about our history and our relations to all other living organisms on Earth. We're all related, in some ways very distantly, and to other organisms not so distantly. But one of the key pieces of evidence is that we all share this unique molecule that contains instructions for building organisms. The structure of DNA has been studied for a long time. It was first examined by Linus Pauling, who had won a Nobel Prize for looking at other molecules, and then expanded upon by Rosalind Franklin and colleagues here in England, who performed something called X-ray crystallography, which really sort of set the framework for further work in elucidating the structure of DNA. And then finally, the double helix molecule and the complementary base pairs were put together by these two guys, James Watson and Francis Crick. One of them was British and one of them worked at Harvard. They ended up winning the Nobel Prize for their work, but most people now recognize that their work simply could not have happened without the work of Rosalind Franklin, who unfortunately died just a few years after her most important work in her early 30s. So a very promising career was cut short. And unfortunately, they do not give posthumous Nobel Prizes, but most people now recognize her as the principal discoverer of the structure of DNA. It should also be noted that we want to give James Watson as little credit as possible because he is a well-recognized raving racist lunatic who continues to spout crazy stuff about all sorts of people. You can look it up on the internet. He's lost all sorts of honors and awards and other such stuff. Of course, you can't take the Nobel Prize away, but at one point he actually offered to sell it because he was losing money. So the guy is an absolute piece of work. So we should give her credit and not him. So let's review. DNA is this double helix molecule with a sugar phosphate backbone. The sugar is deoxyribose. The phosphate group links those sugars, and the two corresponding sugar phosphate backbones are attached to one another via hydrogen bonds that connect any of 4 nitrogen-containing bases: adenine, cytosine, guanine, or thymine. The nitrogen-containing bases bind complementary to only one other base, adenine to thymine and guanine to cytosine, and vice versa, because of their structure and the nature of the hydrogen bonds that can be formed between them. And so this makes it a molecule that is easy to replicate, which is obviously critical for things like cell division, because from one strand you can make an identical copy of that strand. Again, just as review, here are the different bases here. The adenine-thymine base pair here creates 2 hydrogen bonds. The guanine-cytosine pair contains 3 hydrogen bonds. So take-home message, and this is, again, review. DNA is a nucleic acid, a macromolecule that stores information. It consists of individual units called nucleotides, which consist of a sugar, deoxyribose, a phosphate group, and a nitrogen-containing base. DNA structure resembles a twisted ladder, with the sugar and phosphate groups serving as the sides or the backbones of the molecule, and the bases serving as the rungs in the middle. Okay, so we have all these bases, and what do they mean? We have A, C's, T's, and G's, which appear to be in random orders, but we know that's not the case. They're telling us something very important. They're giving us instructions, and those instructions are genes. Genes are just sections of DNA that contain instructions for making proteins. As we said at the very beginning, DNA provides the instructions for building virtually every organism on Earth. And it's important anytime you look at a set of instructions for them to be correct. We know that if you're building a piece of IKEA furniture and you get the wrong instructions for what you're building, you can't build that piece of furniture with the building blocks you have. But another really critical part to DNA is when those instructions get turned on and where they get turned on within a developing organism. So again, if we use the analogy of IKEA instructions, you tend to start at step 1 so that you can do step 2, and then you do step 2 so that you can do step 3. If you just jump ahead to step 6 after step 1, you can run into problems. And if you just kind of willy-nilly try and do it, you can't necessarily put together the structure you're trying to make. And the same, again, is true with the organism, that not only do the instructions need to be correct, not only should there not be mutations in the genes, but very frequently, organisms are put together in a very particular and precise manner, with certain things being put together in certain areas of the developing embryo and only at certain times. So the instructions are critical, but the ordering is just as important. Excuse me. Okay, let's keep going. Now, DNA is contained on chromosomes. Those chromosomes, and again, this is review, are linear in eukaryotes and circular in prokaryotes. Humans have 23 pairs of chromosomes, and we have about 6 billion base pairs in every cell. But that doesn't mean that we have 6 million or 6 billion genes, because genes can be of varying sizes in terms of the number of base pairs that they are. Now, the number of chromosomes varies from species to species, and you might make the assumption that more complex organisms should have more chromosomes. But what really is complexity? It's not something that's very easy to measure. One classic way that complexity is measured is by the number of cell types an organism has. So if you have a lot of cell differentiation, so you have cells specific to the lining of your intestine, and you have nerve cells and brain cells and heart cells. But I'll just tell you right now that fruit flies basically have the same number of cell types as humans, and they only have 4 chromosomes, and humans have 23 or 23 pairs, right? What about goldfish? So a goldfish has 47 chromosomes, or rather 47 pairs, because again, they have a homologous pair for each one. And I would argue that a goldfish is equally as complex as a human. Why do they have twice as many chromosomes? Dogs and chickens have 39 different chromosomes. And any of you that have seen my dog in these videos realizes that that is not the most complex creature. So there's no relationship between the complexity of an organism and the number of chromosomes that that organism has. But what about the number of genes, right? Because you already said, well, okay, maybe they have a lot of chromosomes, but the gene size varies, since you can have different sizes of genes in terms of the number of base pairs. So is there a relationship between complexity and the number of genes an organism has? And the answer to that question is no. The amount of DNA in each cell is known as the C value. And that's actually something that you can measure by simply weighing the chromosomes, weighing the genetic material. So the total weight of DNA in a cell is known as its C value. And we'll get to that in a second. So genes are a sequence of bases in a DNA molecule that carries the information necessary for producing a functional product, usually a protein molecule or an RNA molecule. They average, and again, this is average, about 3,000 base pairs, but they can be much shorter and they can be much, much longer. Now, if we review from yesterday, one of the things that Mendel elucidated was that each parent has 2 copies of every gene, whatever a gene was, and he didn't even know at that point, but that they only give one of those copies to their offspring, and that those copies can be slightly different versions of each other called alleles. And you should remember which law of Mendel's that was. That was Mendel's law of segregation. So Mendel said that each organism has 2 copies of whatever codes for a trait, and those copies can be slightly different versions of one another, like white flowers versus purple flowers. So if genes are the sort of molecular unit of heredity that codes for a particular protein, alleles are just versions of that gene. And again, using flower color down here, we might say that the gene is the area on a chromosome that codes for the color of a particular flower. But depending on the version of that gene or allele, you can get slightly different proteins that produce pink flowers, white flowers, or yellow flowers. So that's something important to remember. So take-home message. DNA is a universal language that provides the instructions for building all the structures of all living organisms. And the full set of DNA an organism carries is called its genome. I hadn't mentioned that before, but I'll say that now. If you combine all the genes in an organism and you ask, what's the genotype of the entire organism? It's its genome. Review: in prokaryotes, the DNA occurs in circular pieces. In eukaryotes, the genome is divided among smaller linear strands of DNA called chromosomes. Finally, a gene is a sequence of bases in a DNA molecule that carries the information necessary for producing a functional product, usually a protein molecule or RNA. And although we haven't gotten into this, I have mentioned elsewhere, although I don't remember where, that proteins aren't necessarily strictly for building things like muscle or bone or anything like that. There are proteins that act like little molecular light switches, and those are going to be very important when we get to the section on gene expression and regulation. What determines whether a gene turns on or turns off, or where a gene turns on or turns off? It turns out that it's proteins that are coded for in your DNA that basically act like little switches telling other genes, okay, time to turn on or time to turn off, depending on the environment that they're in. And that probably doesn't make a lot of sense now, but hopefully it will in a few minutes. I want to get back briefly to this idea that we said there's no correlation between complexity and chromosome number. And then we postulated, well, maybe there's a relationship between the genome size, the number of genes, or the number of base pairs, and complexity? And I told you before that the answer is, well, no. And so here's a graph of that. What you're seeing here are the number or estimated number of base pairs. So this is the genome size. And this is in millions of base pairs. So a fruit fly has 180 million base pairs. A human has about 3.5 billion base pairs. An onion has about 180 million base pairs. A salamander has about 840 million base pairs. 1.8 billion base pairs, 8.4 billion base pairs. An amoeba has 67 billion base pairs of DNA. And I would argue that an amoeba is not more complex than a human because an amoeba is a single-celled organism. So if we're using the number of cell types to determine complexity, well, yeah, this is not a very complex organism. So here's this paradox. An onion has 5 times as much DNA as a human. And this paradox is literally called the C-value paradox, which asks, why do simple organisms sometimes have so much DNA? Now, you might say, well, an onion and a human aren't very closely related. So there could be all sorts of reasons why they have such different amounts of DNA. But here are 2 freshwater fish. These are both sturgeon, so they're very closely related. These are the fish that produce caviar. And there is a 3-fold difference in the number of base pairs just within this group of fish. So something really strange is going on. And a hint that we will answer when we get to our section on evolution as to why there is such disparity has to do not so much with the number of base pairs that there are in a genome, but rather with the proportion of the DNA that codes for genes in the genome. Aha, okay, so not every base pair or sequence of base pairs in a genome codes for a gene. How much does code for genes? Well, in humans, of those 3.4 billion base pairs, only 2%, that's a really small number, code for genes. So only 2% of the bases out of that 3.4 billion are actually useful in making proteins. What about a fruit fly? 20%. What about a roundworm? 25%. What about a plant? 28%. What about a bacteria like E. coli? 90%. So now we're starting to see a pattern that it appears that the more complex the organism, the less percentage-wise of its genome actually codes for genes. What does the rest code for, and why is it there? Well, I'll tell you, the rest codes for nothing. Regions of DNA that produce genes that then produce functional proteins are called coding regions. Regions that do not produce genes are called non-coding regions. And those non-coding regions can take place either in between genes, like what you see right here, or within genes. So here's a gene where there's a section that's coded for, then a blank section, then a section that's coded for, then a blank section. 75% of non-coding DNA is the type that's found between genes, and 25% is the stuff found within genes right here. Now, you may have heard of genomics. Genomics is a whole field of study where you look at an organism's entire genome to elucidate how it works at the cellular level, at the physiological level, at the biochemical level. There's a closely related field called transcriptomics that looks at the transcriptome, and the transcriptome is a subset of the genome whereby you're only looking at the genes themselves as opposed to the blank areas. And there's reasons for why you might want to do that versus looking at the whole genome. But the take-home message of this slide and of the previous slide is that in most eukaryotic organisms, the DNA does not code for any proteins. And again, in humans, only 2% of the total DNA of the 3.4 billion base pairs codes for genes. And that corresponds to about 10,000 genes in the human genome. These non-coding regions of DNA are called introns. I like to think of them as being in between genes, so they're not really coding for anything. The coding regions of genes are called exons, but introns are the non-coding regions. They may take the form of short or really long sequences, and they're often repeated thousands of times. So you might have a section of DNA that is just ACT, 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 over and over and over and over and over again. Introns can, and we'll explain what those are later, but it's kind of intuitive. It's just a portion of a gene. They can refer to duplicate versions of genes, and they can refer to something called pseudogenes. Pseudogenes are genes in the genome that, for one reason or another, are no longer functional. And a good way to think about this is if you just abandon your house and you stop mowing the lawn and washing the windows, like over time it starts to fall into disrepair. Well, when genes are no longer used by organisms for one reason or another, the same thing happens. They don't just disappear from the genome. They're still there, but they kind of start to fall apart. And we call those pseudogenes. So take-home message. Only a small fraction of the DNA in eukaryotic species codes for genes. The function of the rest is still a mystery, though it may play a role in gene regulation, and I'll add something here. It definitely plays a role in evolution, and again, we'll talk more about that in a week or so. Okay, we'll talk about how genes work in the next video.